So what we're going to do now is we're going to go through the short answer of NEEP topic test 1. Hopefully you've had a look at the multiple choice. There's only 4 questions to go through, so bear with me. These things are very interesting and I like to talk about them in a big picture sort of way. They're very relevant throughout the entire course. So let's zoom in and have a look at the first question. Alright, so question 1 is with 8 marks. It's really important that when you're going through short answer that you do take the time to have a look at how many marks they are worth, how they're broken down, so that you can see how many keywords or how many concepts you need to discuss or how many things that you need to compare. Okay, really important that you're using that as a tick list for yourself, along with also using the 8 minutes for the 8 marks. So remember, a mark per minute. It's really important that you're starting to use those strategies. So plasma membranes are described as a fluid mosaic made of structural components which also have receptors on the surface. So we have talked about this fluid mosaic model in class before, and pretty much what it is that if you remember talking about when you make a bubble, so if you're doing the dishes or even if you're at a wedding or outside and you're making all those bubbles and you have a look at those bubbles and they're kind of moving, that's exactly what's happening to the cell membrane. So the phospholipids are not concreted in, they're moving around a lot. Okay, so I'm just going to move over for a sec. Ooh, where are you? So these phospholipids that you can see and the protein channels and the cholesterol and the glycoprotein, they are all moving around. Okay, they're not in that permanent spot. So that's what it's meant by a fluid mosaic model. And the reason why we say it's a mosaic is because it's like, if you're looking at it from above, it's like we've got all these little tiles that are making up a big picture. Okay, so you might be asked to explain that in an exam, so that's why I just went through that. So the last bit of the prompt says, which also have receptors on the surface. All right, I'm really hoping that because we've gone through the multiple choice, is that you know that a receptor is a type of protein. Okay, proteins are really important. They rule the world. Beyonce should sing about them. So I just want to go back to what I was talking about in the multiple choice section, how I said that there was 2 different types of hormones. All right, so we had, hopefully you remember, the, need a pen, peptide hormone, all right, which is made out of protein. That one's pretty easy to remember because polypeptide is protein. And then your steroid hormones, which, by the way, are all your sex hormones, are lipid hormones. Okay. Now, you'll know that lipids are non-polar. And they're also hydrophobic. They're not big fans of water. But that means, oh, I don't want that, go away. That means, though, that they are able to dissolve in lipids. What's going on? Okay, so the membrane itself is, remember, it's composed of a majority of phospholipids, all right? So if I have a hormone that is made up of lipids, it's going to be able to move straight through because like dissolves like. Like dissolves like, really important concept. So that just means that this steroid hormone, which we know is a lipid, is going to be able to diffuse through or move through that lipid membrane because it is non-polar and hydrophobic, the same properties as that membrane. Okay, so the receptor for that will be inside the cell. Whereas if we have a look at these peptide hormones, okay, they're proteins, which means that they are hydrophilic, so they like water, but remember lipids don't like water because they're hydrophobic. So they're not going to be able to diffuse through as easy. So what we need is something called a receptor or a transmembrane receptor. So remember the word trans just means to go across. So it's going across the membrane. So we can see that we've got 3 different types of examples, and we'll go through this more in Unit 3. Unit 3. But the 3 different types, I just want to make you aware of them, is we've got the G protein, okay, so that sits through there, and the ligand here is just, we see that in all 3 examples at the top, it's just a word for a small molecule, like a hormone, okay, so not all chemical messengers are hormones, but majority of the examples that we do look at in biology year 12 are hormones, all right, so you might need that hormone or that ligand to come and open a protein channel in order to let something through. Okay, so that's acting as a receptor. That's triggering the opening. In this case, we call it a ligand-gated ion channel. All right, and that's opening the channel. So therefore, something that was on the outside can now move onto the inside of the cell. Okay, this one here, the receptor tyrosine kinase, there's an enzyme on this side, so therefore it might be causing a different reaction on the inside of the cell because of this chemical messenger here, and the same with the G protein. All right, but I really want you to focus on the next part of the question, this example here. All right, so the ligand-gated ion channel. So the receptor on the outside, the transmembrane receptor, will receive the signal from the ligand. And remember that everything in biology is specific to its structure in order for it to carry out its function. So that ligand or that hormone will be a particular shape in order to bind with the receptor that's on the membrane. Okay? So it's important that you need to know. All right, let's go back to the question. I seem to do this a lot, don't I? Okay, so that's receptors. So remember, just keep in mind that receptor is protein. Okay, the receptor can either be on the inside or the outside of the cell, depending on the properties of the signal messenger, and that will induce a response. Okay. All right, so let's go on to A. So draw and label the 2 main structural components of a plasma membrane. All right. So hopefully you recognise this diagram here from the classroom. So it's asking for 2 main components. I think that there might be a few more than 2 that you need to include, so I'm just going to go through them now. So this structure here is obviously a phospholipid. Okay, but if you just call it a phospholipid, I don't think that's enough. So what you need to do is break it down. So we've got our head here. Oh, that's not how you spell head. Better, and then you've got your tail. But it's really important that you are describing the properties of those heads and the tails of the phospholipid. Okay, so I want you to think that there would be fluid here and there would be fluid here. So both of the heads are touching the fluid, so that must mean that they are hydrophilic. They like water. Okay, whereas the tails, they are in the inside, so there's not as much fluid there, so they're hydrophobic. So when you are labeling a diagram, you must say the hydrophilic head and the hydrophobic tail. Okay, if you don't add those in, you are not going to get the marks. All right. The other things that you might include in your labeling is your cholesterol. Cholesterol. So that's important in ensuring that there's less rigidity within the cell, so that these tails that are adjacent to each other don't start to bind together and fuse. Okay, so that's what allows us to have that mosaic fluid model, which is the cholesterol. Okay, and the other thing that you might want to label, I'm just going to change colour, we'll go with this purple, is you might have protein channels or protein receptors, or okay. But I think that that's important that you label that as well. So if you go back and you have a look, it's saying to you that it's worth 2 marks. Okay, so most of the time, if you just label the hydrophobic head and the hydrophilic tail, that will give you enough, but you never know. So label as much as you possibly can when you're in an exam. Beautiful. So let's move on to question B. This is why I was rambling on before. So describe a function of the receptor molecules on the plasma membrane during the movement of molecules through the membrane. Okay, so receptor molecules and their importance of things that are moving through the membrane. Now, before, if I hadn't talked about receptors, you might have just thought that we're talking about facilitated transport, and you might have just said that they would move through from a high to a low concentration. All right, but in this case, we're talking about a receptor that might have not been open or permeable to that substance in the first place. Okay, so we're going to go back here and have a look. The ligand or the hormone will bind to the receptor. So remember, the receptor is a transmembrane receptor. It's all the way through here. We can see that in the red. So that's going to bind to the ligand, and now that because it's received that signal, that's now going to open, and then the substance can then move through, which we can see with the arrow there. Okay, so when we go back to this question B, the sorts of things that I would like you to write is probably even use an example. So we're going to go back to the insulin example. So the receptor detects a chemical messenger or stimulus, in this case, which would be the insulin, which would then provoke a response, which would be the opening of the channels and allowing the substance to enter via facilitated diffusion. Now that seems a lot, doesn't it? But I'm going to try and simplify that for you. And that's not what I want to do. What we'll do over here, so the stimulus response model, I think it's really important that you're aware of that. So we have the stimulus, and then we have our response. Okay, so the initial stimulus would be the insulin in this case, which is the chemical messenger. Now remember, this is a protein. All right, it's a hormone, but it's a protein, so it's a peptide hormone. All right, now that's going to bind to the receptor, that transmembrane receptor. Okay, and then the effect will be that it's going to open, and the response will be that glucose can then move through. Because remember, insulin wants to skip black. The whole role of insulin is to lower blood glucose level. Okay, and the way that it does that is it binds with this receptor that's a transmembrane receptor. Okay, which are usually found on skeletal muscles. And then that binds with the receptor, which then causes that to open, which then the response is that glucose then can move freely into the cell via facilitated diffusion. Okay? Facilitated. Diffusion. Alright? I know that's a little bit messy. You'll be alright. Okay, so that's 2 marks. So I would really make sure that you're talking about the stimulus and the response. Okay, always go back to that stimulus response model. That's really important. Okay, beautiful. Let's move on to C. Okay, so complete the table of the following table, comparing the movement of water to the uptake of glucose by a cell. All right. So So water, we know the process for water moving across is osmosis. So that's something that we can definitely write in there straight away. Okay, active or passive, does it require energy or not? Okay, the movement of water is always going to be passive. All right, the way in which water moves through. Now, I would like you to be more specific here, and I'll just try and zoom in so I can write it a bit bigger. Okay. Is I would like you to write aquaporons, because water as you know, is polar, is hydrophilic. It loves itself, obviously. And the lipids are hydrophobic. They don't really like water. So a lot of students come to me and say, Miss, why would water be moving through the phospholipids when it doesn't, they repel each other? So there is a type of protein channel which we call aquaporins, and that's where the water generally will move through. If it really dire, like it's very dire and needs to get through, then it will move through the phospholipids, but it doesn't like to. Okay, so both responses would be correct, but your aquaporins, I think, would probably be a better response to write. Okay, so your glucose then. So remember, glucose is a small molecule in terms of when we're looking at carbohydrates, but if you're looking at an overall sort of picture of it, it's quite large. Okay, so remember when we were having a look at this diagram, it's gone. This diagram here in class, and we were having a look at where carbon dioxide and oxygen and water moves through. So carbon dioxide, and remember that's CO2 or even oxygen, O2, they're able to move through in between the phospholipids, okay? And remember, glucose, still think it's small, but it's C6H12O6, okay? In retrospect, to the carbon dioxide and the oxygen, this is quite large. So what it's going to need to do is it's going to need to go through a protein channel, which therefore is facilitated diffusion. Okay? All right. Get the pen. So write that in here. After. So this will definitely be through the protein channels because it is too large. A lot of things actually move through the protein channels. Only little tiny things such as gases or will move in and out through the phospholipids itself. Okay, so when we're drawing cells, actually, we're really simplifying them. There should be a lot more protein channels. All right, coming to the active and processed passive. So it can be either active or it can be passive. So when it's passive, we just refer to that as facilitated diffusion because facilitated, it's requiring help. Oh God, that looks awful, sorry. Facilitated diffusion. If, however, let's change colour just so we can see that this is a difference. If it's active, and I'm just going to write it down here, we would refer to then this as active transport. This is a really big common misconception that students get wrong. They think that active transport and facilitated transport are the same thing because they're using the protein channel. Facilitated diffusion is when it's going with the concentration gradient, so it's going to go from a high to a low. When we're using active, it's going against the concentration gradient. In both cases, glucose is always going to use the protein channel because it's a large molecule and it's not going to be able to fit through the phospholipids, but you definitely need to know that one's going against the concentration gradient, one's going down the concentration gradient. All right, last component for this question. Yay, so you can stop talking, you're thinking. So D, what is the role of cholesterol in the plasma membrane? Well, if you were already listening, you would know this. Okay, so it's to reduce the rigidity. I really like that word, rigidity. Okay, so it reduces its rigidity, or it's less rigid, or it's more fluid, something along the lines of that, and one of those will just give you the right amount because there's only one mark. Okay, beautiful. Okay, so now we're going to move on to question 2. Hopefully this one's a nice quick one. So it's worth 6 months again, so you should be giving yourself 6 minutes to do it. So DNA is a double-stranded helical molecule, so that just means that helix just means that just like you see in a helicopter, it means that it goes around. Okay, the basis of one strand determine which polypeptide will be synthesized. So break that down. So polypeptide, you know that means protein. okay? And you mean synthesize, you know, synthesize means to create. All right, and you know that one strand, so you know that DNA has 2 strands, so it looks like this. So one of those strands, either the blue or the pink, is used in order to create a protein, okay, a polypeptide chain. All right, so what is the subunit of nucleic acid? So this is just coming back to what is the monomer of a nucleic acid, and that one's really easy. You know that one's a nucleotide. Okay, it's almost a free mark. Okay, double I. Using the following components, illustrate the subunit in the space below. Again, really simple. So what I'm going to do is I'm just going to move it up, get a bit bigger. Oh, shaky. So nucleic acids, remember, there is 2 different types. There's DNA and RNA. So I want to just go through it and compare it with this. All right. So in the middle of your nucleotide, there should always be your pentose sugar. Okay. Then connected to that will be your nitrogenous base, and then on the other side will be your pentose sugar. Okay. Now what we need to do is go through. So in your pentose sugar, there's going to be 5 carbons. Okay. Hence why it looks like a pentagon. So I've called it pentose sugar. Okay. On the first carbon, we have our nitrogenous base. On the second carbon, we have generally what I'll go for here is we have an OH group. Okay. On the third carbon, we also have another OH group, and then on the fourth carbon, there's an O here. So that's not actually a carbon at the tip. And on the 5th carbon, there's your your phosphate group. Okay. Now I want you to pay close attention to this one here. So your second carbon with your OH group. So the only difference between a nucleotide from a DNA and a nucleotide from an RNA is to do this OH group. Okay. So if you have a look at DNA, it's called deoxyribonucleic acid. Okay. Oxy is referring to this oxygen group here that's in your ribose sugar. Okay. So the D that's here refers to a missing oxygen. So in DNA, on its nucleotides, it only has a hydrogen. Okay, whereas on a RNA, it is called a ribose nucleic acid because it doesn't have an oxygen missing. So its group will look like this. All right. When we have a look at DNA further in the course, it'll ask you'll need to know about 3, 5, and 5 prime end. This is really easy. I've labeled the carbons for you so you can still see. So the 5 prime end is where the 5th carbon is. So the where the phosphate group is. Okay, and then your 3 prime end is over here. Okay, so if you need to label a nucleotide, that's your 5 prime end, and this is your 3 prime end. Okay, so that is really important when we start looking at DNA replication and transcription. All right, so that's important to know. So the only other things that we need to touch base on with DNA, the other differences is that DNA as A and T, and C and G, and here comes my dad joke, so watch out. DNA has A and T, it's A and U because it's AU. Why did you change for? So it's A to U, but it still also has C and G. DNA is a double-stranded molecule, whereas RNA is a single-stranded molecule as well. Okay, so hopefully that helps to clear things up. All right, let's go on to the next part of the question. So the template strand is copied. So remember how we read at the top, it said that only one strand determines which polypeptide will be synthesized. That's referring to the template strand. Okay, so one strand is called the template strand, the other is called the non-template strand, or the coding and the non-coding. All right, but we'll go through that a little bit more when we go to Unit 4. And the product dictates which amino acids are linked together at the ribosome. So remember, when you're, so at the ribosome, so we need to have, it goes from DNA to mRNA, and then that goes to the ribosome, and then that produces your protein. Okay, this process, or it goes from DNA to mRNA, that occurs in the nucleus, and that's called transcription. I apologise if you can hear Mavi barking. Okay, so that's transcription. When we are looking at the protein actually being transcribed, and that happens at the ribosome, we call that translation. Okay. So, what is the process called when the template strand is copied? All right. So, meaning that it's forming the mRNA. So that is called transcription. Okay. What is the product? We've already gone through that. mRNA. Now, when it comes to abbreviating things, I'm pretty sure I've mentioned this in class as well. So when you're looking at organelles, if you're talking about the rough ER, you would never write rough ER. You would write rough endoplasmic reticulum. The same here with your mRNA. I would encourage you to write messenger RNA, simply because sometimes the examiners feel that they need to do that in order to differentiate between students. So make sure you're doing that. Okay, and then the lucky last one for this one is state two differences between RNA and DNA. We've already done that. So we know that, and I might just move this over. So we've got a new space. I can make more room. So DNA, RNA. Okay. So we know that this is double-stranded, whereas RNA is single-stranded. If we have a look again at that second carbon, we know that the DNA is missing one, so it's only OH, and that's why we refer to it as a deoxyribose sugar. Okay. Whereas the RNA has that OH group still. So we can still refer to that as a ribose sugar. So the deoxy, D just refers to it's being removed, and the oxy being that's removed the oxygen. Okay. We know that the bases that we see in DNA is A and T, with C and G, and we know that my dad joke is AU. Why did you change? So A is now base pairing with U, and C is base pairing with G. Okay. Beautiful. Ooh. Awesome. So I think that's question 2 done. Let's move on to 3. Move away. Okay. So hemoglobin, which transports oxygen around the body. So remember, before we even read the next question, hemoglobin, I was telling you before that this is an example of a protein that has a quaternary structure. And a quaternary structure is one that has 2 or more polypeptide chains. Okay, so remember, it's not proteins, it's polypeptide chains. So the hemoglobin, which transports oxygen around the body, consists of 2 alpha and 2 beta chains of amino acids. Which type of protein is a hemoglobin in terms of its structure? So this is asking you, what structure does this protein go up to? Okay, and so remember, it's the quaternary structure. Okay. That's what that's asking you there for. All right. Explain how a change in the DNA could lead to a non-dysfunctional hemoglobin. So that just means that how will we go from DNA to the protein, which is a hemoglobin, and how would there be any errors that occurred in there? So I do have a diagram here that I want to go through. Oh, it's here. Okay, you've all seen this on the back wall. So we're just going to go through a bit of transcription translation. Okay, so this is the nucleus. You can see that there. This is our DNA that we've got here. And this is inside the nucleus. So the DNA can't leave the nucleus. So what happens is that we go through a process called transcription, which you can see here, and that creates your DNA to a portable messenger called your messenger RNA. All right. Now that leaves through nuclear pores. All right. And then that goes to the ribosome. The way in which that is read at the ribosome is in something called a codon. Okay. Now, if you're a big Disney fan, you know that everything happens in threes and it's a lucky number. It just happens to also be my birthday. So the mRNA is read in groups of 3, and these groups of 3 are referred to as a codon. So I'm just going to write up here that it's got 3 base pairs that it's read in. Okay, so it goes in and that's read in here by the ribosome. Now when that's read by the ribosome, there is a corresponding almost matching codon, which we refer to as an anticodon. Now those anticodons are found on the tRNA. So that's these green things that are floating around. Okay, so the codon, when that's read by the ribosome, if a matching tRNA molecule comes along and they bind together, they're complementary to each other. Okay, see how this tRNA here on the top has like a little red molecule? That's an amino acid. So if they're complementary to each other, they will bind together and that will leave an amino acid. And you can see here that we've got the polypeptide chain. Okay, so it's starting to form there. And that's how a protein is made. So remember that the order of amino acids is the primary. So the order, I'm just going to write a P because I don't have any more space. Okay, and then the secondary is your alpha helix coils. I'm going to run out of room too, so I'm just going to write alpha and then beta for your beta pleated sheets. Okay, and that's just secondary. Your tertiary, remember, you need your chaperone proteins. Okay, which gives you your functional 3D shape, and if it was an enzyme that we were looking at, it would give it its active site. Okay, so let's go back to the question. Explain how a change in the DNA could lead to a non-functional hemoglobin. All right, so remember that DNA codes for proteins, and that the DNA codes for the protein by coding for the sequences of the amino acids. Okay, so when the ribosome is reading the codons, so the groups of the 3 base pairs, that is then complementary to the anticodon that's on the tRNA, and then that drops off in an amino acid, which starts the polypeptide chain, which is the primary structure for the protein. Okay, if there are any changes within the DNA, so this can occur, so we refer to this as mutation. So I might just change a coloured pen and write that there for you. So mutations, and we will look at these a lot more in detail in Unit 4. So if there's any sort of mutation, so any changes to either the DNA or the mRNA, the way that it's read at the ribosome can be different. So if there's any changes to that codon, it might actually be complementary to a different tRNA. And all these tRNAs actually have a different type of amino acid on the top. So if they code for a different type of amino acid, then there'll be changes within this polypeptide chain, which means that therefore it might not have the correct primary structure, therefore it might not have the correct secondary structure, and therefore the tertiary structure might not be able to form in the correct way for it to do its function. Because remember, structure is extremely important for function. All right, so what you need to write down there on your answer is that something along the lines that DNA codes for proteins for the sequence of amino acids, and if this is altered in any way, then it may translate for a different amino acid, which might therefore change the secondary and therefore the tertiary structure, which is the functional shape of a protein. Okay, so hopefully that helps. I might actually just go back and write that there for you because I think that's a lot of information to take in. Okay, so DNA, I'm just going to put it in shorthand for you. DNA codes for codes for the amino acid sequence. If this is altered, this might change. Therefore, it might alter the primary, the secondary, and then the tertiary level. And the tertiary level is its functional shape. Okay, so therefore, it might be non-functional if this tertiary level is affected. Okay, hopefully that clears that up. Oh, I love these questions. Okay, C, name the components X and Y. I think they're pretty easy, and hopefully by now that you are recognizing that as being a protein. Okay, so I'm just going to go over it. So X is here. All right, they look like coils to me, so remember, alpha, helix, coils. Ensure that you have full names. And then this is going up and down. You see another one here. Okay, so it's beta, pleated sheets. Okay, so that's quite easy. So that was 2 marks for that, so you would have got 1 mark each. All right, and then it says for double I, what physical features do these components give to the structure? So I just want to go through this with you. So the beta plated sheets give it strength. All right, and then you guessed it, the alpha helix coils gives it flexibility. Pretty straightforward, I think. Okay, name one structure or part of the human body which contains secondary structural proteins. Okay, now we mentioned before that they're probably going to be in the protein channels because most of the time they are just a secondary level. Okay, but some other things that you might have seen might be in your skin or your nails or your hair. Okay, so keratine's a really good example of that. Beautiful. Almost done. 1 more to go. So lucky last question with number 4. So it's worth 9 marks, so it's quite a significant amount. Again, a diagram here, so please make sure that you are always annotating your diagram before you get started, all right? Because sometimes you might look at a diagram, feel really confident with it, and then you don't annotate it, and then you go to read it, and then it confuses you, and then what you thought you knew, you've now muddled up, all right? So that's my approach with it, okay? So let's start with what we know. So this line here looks like it must be the membrane. Something's going on here, so maybe I'm going to say that exocytosis is occurring because it looks like there's maybe a vesicle that's moved towards that membrane, and I reckon that maybe A is that vesicle. And if we're looking at vesicles and exocytosis, maybe I'm even thinking that we've got maybe some protein synthesis that's going on. So I haven't read anything else just yet. I reckon this looks like a mitochondria. And whenever I see a mitochondria, I'm always going to think of energy. And then I might even think about an active process, which exocytosis does require energy because it's bulk transport. Okay. What else have I got here? I can see that must be the nucleus. And I've got some genetic material in there. I'm running out of room though, so I'm not going to write that there. Let's get a different coloured pen. Then B, it looks like we've got a ribosome. I think that looks, because it's pointing just at the black dot. And then on G, it looks like that must be the rough endoplasmic reticulum. Now, I'm only going to write rough ER. However, like I said before, if you can write full words, I really encourage that you do that, because sometimes the examiners won't give you full marks for that. I'm probably even going to write rough ER for that. E we've labelled as the vesicle. Now, E looks a little bit funny. I'm not gonna lie, that looks a bit strange. But again, try and use your understanding of the cell when its makeup here. So you've got your nucleus, and then it's not the colour I wanted. You've got your nucleus, then you've got your ER that's attached here, then you've got a vesicle somewhere around near the rough endoplasmic reticulum. So E, just from your understanding of protein synthesis, must be the Golgi apparatus. Okay, and again, I'm going to simplify, but please make sure you're writing full names. So the Golgi apparatus can be referred to as the Golgi apparatus, the Golgi bodies, but never just the Golgi. Okay. So what else I'm going to annotate on here is maybe the order in which protein synthesis occurs. So I know that the DNA within the nucleus needs to get out, and so that needs to happen, needs to turn into an mRNA. So that's going to be transcription, because remember, you've got to write the play before you can translate it. So transcription. Then that's going to go, that mRNA is going to go out into the rough endoplasmic reticulum, okay? And at the ribosome, we're going to have translation. And so remember, at the ribosome translation, the mRNA is going to be read in groups of 3 base pairs, which is going to be the codon, and then that's going to be complementary to the anticodon that's on the tRNA. And when those complementary codon and anticodon come together, then an amino acid will be dropped off, and so forth and so forth. So therefore, one amino acid, another amino acid, another amino acid, which are joined by a peptide bond, will form the primary structure of a protein. And then because of the interactions with the R groups, then the secondary structures, so the alpha helix coils and the beta pleated sheets and the random coils will start to form. They will start to form due to those attractions, also the tertiary structure, but then we need to have our chaperone proteins to come in and finalize that. And remember that the protein functional shape is at the tertiary level. So if we're looking at enzymes, the tertiary shape will be the active site. Okay, but I'm getting off track now. So once the protein is made through translation at the ribosome, then that's because it's going to be an extracellular protein because it's made at a ribosome on the rough endoplasmic reticulum. That's then going to be sent to the Golgi body. Okay, the Golgi body is then going to modify and package that protein, which is then going to then form a vesicle, and then that vesicle will then start to make its way to the plasma membrane where exocytosis occurs, and it binds with the membrane, and then that protein is then free. Okay, beautiful. So now knowing that, let's have a look at the table below. So again, I know that you probably know that, but it's important to do before you look at the question. So they've done the first part for you, so actually I'll read the instructions. In the table below, name the organelles labelled in the diagram above and give their function in biosynthesis. So remember when we had a look at this word biosynthesis before, bio meaning life, and we know that protein is extremely important for life, okay? because enzymes do so much. And synthesis means to create. Okay? So we're on the right track by when we were having a look at the diagram and brainstorming about what the question might be. So A, we were correct, is a vesicle. All right? Contains synthesized material. So in this case, the protein. B, so let's have a look at that. I'm just going to change my color. Oh, yep, that's here. B is your ribosome, okay? And that's the location of protein synthesis, so translation. Okay, so either of that would be acceptable. D, they've done that for you. Mitochondria provides ATP for the process. And then in this case would be exocytosis because of bulk transport. All right. E, let's have a look. That's here. Okay, beautiful. That is the Golgi apparatus or the Golgi bodies. Remember, you can write either. And that packages and modifies the protein and puts it into a vesicle. All right. And then lastly for this one, G, which is over here, is the rough endoplasmic reticulum. Remember, please, to write full words, not rough ER, not ER, must be rough endoplasmic reticulum. Okay, and the function for that is that it's is the place where proteins are modified and synthesized. Okay, so they're made, created, and then packaged off. All right, beautiful. Let's just move down a little bit. Cool. So now we're on to B. Name the process occurring at F. All right. So because, again, that we've already annotated this, that we know that that's a vesicle and that's trying to move out of the cell. So that's exocytosis. Okay. So I'll just write that in there for you. Exocytosis. Okay. C. In what form is the instruction for synthesis carried to the organelle B? Okay, so the instructions, so whenever you think of instructions, I want you to think of genetic material. All right, so inside the nucleus, we have DNA, but does DNA ever leave the nucleus? No, because it's too large. So it leaves the nucleus in a form called mRNA. Okay, remember that single strand, and that's able to leave through the pores. So let's read the question again. In what form is the instruction for synthesis carried out to organelle B? Organelle B is the ribosome. And remember, when it goes through translation, it goes from mRNA to protein. So the instruction is in the form of mRNA. So again, try and write messenger RNA, just to ensure the examiner is correct in thinking that you know what you're talking about. All right, and D, lucky last one. What is the chemical component of the dense body with an organelle C? Okay, let's have a look. Organelle C is the nucleus. The chemical component, okay, of dense body within organelle C. All right, so that is your nucleic acid. Okay, so nucleic acid is your DNA. Okay, and then you could have that you've also got rRNA, which is still a nucleic acid. So you've got your DNA, which is in your nucleus, and then your rRNA, which is in the